Hello friend. So in this particular module, we are going to see how a pharmacokinetic or bioavailability studies are being carried out for a new developed product or a new formulation. So what is the rationale for developing a new formulation and how a bioavailability or PK study is being carried out? We are going to see in this particular module. So why new formulations are being developed? New formulation in the sense there are some formulations which is being given for a particular indication for twice daily or thrice daily. Then a company is trying to develop a once daily formulation for the particular dosage form. Then maybe some other alternative dosage forms like capsules or tablets will be developed with the lower dose against the higher dose. So they can go for the nanoparticles or some kind of particle size reduction of the existing dosage form and they can reduce the dose. So these are some of the attempts developed or being carried out by the pharmaceutical companies. So why this is kind of developments happens in the generic formulation industry? The purpose is for the life extension of the products. Like you are for the immediate release products, you are bringing the modified release, extended release, orally disintegrating tablets, something like that. You are bringing new technology to the products for the life extension basis. And the technology can be matrix, osmotic technology, gas returning technology, anything can be used to do the life extension. Or you can introduce a new route of administration. For oral, you can go for the transdermal patches or injection, etc. And there can be a substitution products for the low cost or to make the intellectual property. Like for example, I told you can go for the nanoparticles where you can reduce the dose of the product and you can create intellectual property out of it. And you can go for a new combinations where the products are being separately taken, but you are going to fix those combination products. And then sometimes the active metabolites of the particular product will become a separate product or a enantiomer. For lansoprazole, later dexlansoprazole has been introduced. For paliperidone also, yeah, it's one of the metabolite of the another drug. So these are all the things are being done to do the life extension of the products. So when you do develop these products, you need to understand the pharmacokinetics of the product. So for that, the bioavailability studies against the standard product, that means the existing product, will be carried out. So what kind of life extension products are being developed across the industry? So that we have seen in the previous slide also, and some of the things includes the replacing existing IR formulations for the twice daily to once daily formulation. Then you are making a gas alternative formulation or a site specific delivery of the formulation or fixed dose combinations. So these kind of things are being attempted by the pharmaceutical industry. So this is a typical example how a new dosage form is being developed and what is the advantages of it. So metformin was introduced as an immediate release formulation with 500 mg and it has been administered with 2 to 3 times a day. So then extended release products were developed for metformin with once daily administration. Then later, there is once daily formulation with osmotic technology also developed and introduced separately at the different brand. So this is how the life cycle of the particular drug has been extended. And these products will improve the patient compliance, will improve the safety without compromising the efficacy, and sometimes it can improve the efficacy also. And there can be a life extension of the product, continuous business of the product, and you can generate intellectual property out of the product. And you can definitely achieve a low cost through some other approaches which can be useful for the patient population. So in a simple way, how a modified release product is being developed against a immediate release product, we can follow simplified procedures like assessment of immediate release product for PK profile, then prediction of multiple dose profile if possible through the theoretical assessment. Then theoretical prediction of required in vivo profile of modified release formulation using PK parameters of the IR product. So we have to predict the in vivo profile of the modified release formulation using the PK parameters of the immediate release product by method of superposition or simulation techniques which is available in the various softwares like including GastroPlus and some other softwares nowadays. And we have to calculate the dose required for the modified release dosage form. Otherwise, you can do a practical assessment of target in vivo profile with the dose selection usually twice or thrice of the IR. For example, we have seen the metformin 500 mg. You can go for once a day instead of twice a daily. You can go for once a daily modified release with 1 gram of the dose, or you can increase the dose little higher to save the to maintain the efficacy out of the modified release dosage form. To achieve the target in vivo release profile, design target in vitro release profile. So, in case if you are going, this is what the in vivo release profile you are expecting, then to achieve that in vivo profile, you go for a in vivo target release profile for which you are going to do the development trials of the product. Then you develop a prototype modified release profile with the target release profile. So now we have the modified release formulation, again the immediate release formulation with the target release profile. So now at this stage we have to go for the pharmacokinetic studies with the target release profile, whether the target release profile is going to give the expected in vivo release profile, absorption profile, or pharmacokinetic profile, which is good for the product. That's what we are going to see in the PK studies. So this is a typical multiple dose of the product. The blue line, thick blue line, is the 4 times a day it is being given with the multiple administration in a given day. So every 6 hours this product is being administered because of the short half-life and the short action, pharmacological action. So against this, either you can target to develop a once daily formulation, or you can target to develop a twice daily formulation, so that you are reducing the patient compliance, or you can reduce the cost of the treatment also. So, and you can have a better safety profile by maintaining the concentration in the therapeutic range. So all this can be achieved by developing a modified release formulation of twice daily or once daily against the 4 times daily administration. So for to develop this is the theoretical expected profile out of your existing products pharmacokinetic information. And to achieve this theoretical in vivo profile, you need to have a in vitro target release profile. So the in vitro target release profile is like this. So this is the in vitro target release profile, which based on the simulation you can, based on the development trials you can design. To achieve this target release profile, you will develop different products, and one product, probably one of the prototype you can select it, which one is going to give the typical target release profile. If this is the target release profile, then one product goes out of the target and one product is close to the target, which you can go for the select for the in vivo study. So here is some of the references are given how to design target in vitro in vivo profiles by using the method of superposition. So these references can be useful to do your research in either academics or in the industry. So based on this theory, what we have explained earlier, so here is a case example. For example, I have developed a modified release dosage form of metformin 1000 mg against the immediate release dosage form of metformin 500 mg administered twice daily. So I am going to use one or two prototypes for the study, and in the healthy subjects, I am going to do the bioavailability or for assess the pharmacokinetics, maybe 4 to 8 subjects in the as it is a proof of concept. Then I am going to assess the profiles. So after the study, I'm going to get this kind of profiles. So the metformin immediate release product is being administered twice daily, and it is going to give you a peak like this, a profile like this, and my once daily formulation going to provide the peak like this. So this is the likely scenario when you, after the completion of the study, when you test the two products, and this product is maybe in the expected target in vivo release profile range, because the Cmax is maintained and it is in the therapeutic range, and probably you can see that from 1 hour to almost 10 to 12 hours the plasma concentration is in the therapeutic range, and this may be sufficient to maintain the activity for the 24 hours, and this may be your target release profile, and your prototype may be successful in the bigger studies. So this is how the modified release or formulations are being developed, and this is how the pharmacokinetics are being studied in the during the development. So this particular plot showing a once daily dosage form against the 3 times daily administration of a dosage form. The pink line indicates the 3 times dosage administration, and the blue line administers the once daily administration, and this is also a maybe achieving the target release profile and giving a pharmacological activity throughout 24 hours. This particular slide explaining the twice daily administration of a modified release product against 4 times administration of a immediate release product at a steady state. So here the advantages of the modified release product is that the levels are more constrained or more the range is very narrow from the maximum to minimum concentration. The range is very narrow, and for the immediate release it is goes with a very high concentration beyond the therapeutic concentration maybe, and at the lower level also it may be beyond the therapeutic concentration. But in case of modified release formulation, it is very well maintained within the therapeutic range, and the levels of fluctuation is very less with the modified release product. So this is how at steady state the ideally the plasma profile is ideally expected to reduce the fluctuation of the immediate release product. So once you develop the modified release product and do the pharmacokinetic evaluation primarily as a proof of concept, then you can do further development with assessing the multiple dose pharmacokinetic profile. You can predict also, you can assess also, then choose the best prototype and conduct a single dose and multiple dose bioavailability studies. And you may go for phase 3 clinical trials for the newly developed dosage form for the safety and efficacy of the product. With this, we have come to a conclusion or the end that how a modified release product or a new dosage form is developed, how primarily it is evaluated for the bioavailability or pharmacokinetics of the product, and how it is taken for the further development. So we have seen all these aspects of the new product development. And also we have come to the end that the entire process of bioequivalence, right from the basic requirements to the each and every individual activity like clinical phase, bioanalytical phase, pharmacokinetic phase, and the report phase, then the regulatory and the ethical committee requirements, etc. And with this, we have come to the conclusion of the bioequivalence modules. In case if you have selected the bioequivalence industry as your career industry, I wish you good luck and thank you very much for patience listening.